Hi friends, so we're on to 15.6 stages of translation. We're going to start out with the learning outcomes. We'll talk about the 3 different stages of translation. You should be able to describe the steps that occur during each of those stages, so initiation, elongation, and termination stages. And we'll talk about some of the differences that you might see between bacterial and eukaryotic translation. In general, we tend to think of those eukaryotic organisms a little bit more complex, and so there may be more factors involved. And then we have vocabulary along with that. So the stages of translation are 3 stages. We have initiation, so the starting of the translation, and in general we often think about that start codon, so keep in mind that generally we're looking at that AUG that we would see on the mRNA. We're obviously going to have an elongation period where we have a growing polypeptide, and then at the end of that mRNA there should be a termination code which lets the ribosome know that this is termination, which is the end of the synthesis of the protein, which then can be released. And we can kind of look at it here in this particular picture. So when we look at this, we have the large subunit of a ribosome and the small subunit of a ribosome. What we generally would see is that we have, in this case, this mRNA, right? So we have the 5' end, that start codon somewhere near the 5' end. We have all the codons that are going to come into play for making that protein, and then eventually we're going to have a stop codon closer to the 3' end. The other player that we would imagine in this would be that tRNA, right? That first tRNA that generally we think of as having methionine. So as we think about these different parts to it, we can imagine that we would start with initiation. And in this case, those ribosomal subunits will come together to form a complete ribosome. We will see that that start codon will be recognized and that we will have the tRNA with the anticodon that is complementary to our start codon. And that is the way that we would see the start. Notice that that tRNA has been charged, right? An amino acid specific to that anticodon has been added. And this would happen in the middle of the 3 sites on the ribosome. Keep in mind that there is an E site. I often refer to this as the exit site. We have the P site. You'll see it often referred to as the peptidal. I often call it the peptide because that's where we see that peptide start to grow. And then we have the A site. The A site is where we generally, except for the very first one, generally the tRNAs will be brought in and they will be carrying amino acids. So this is the amino site. So this is that initiation, right? Once it gets initiated and we start with that first tRNA, what can happen is that in the A site, the next tRNA with an anticodon for the next spot on the mRNA will come in that's complementary. We will see a transferring of the amino acid or amino acids here to this new amino acid. Once this tRNA in the P site is no longer charged, right? It's lost its amino acid. It will move into the E site and be released to go out and be recharged. And the tRNA here in the A site that has all of the peptides, all of the amino acids attached to it, will move into the P site and we will just repeat that. This actually then will be terminated as this is being fed through from the 5' being read towards the 3' direction. Eventually we will get a stop codon that will come through. At the point of the stop codon here that comes through, it does not have a charged tRNA that has an anticodon to that. What we have is a release factor that will move in. It mimics a tRNA, but because it has no amino acid, it basically tells the ribosome this is time for the process to end. That means that the polypeptide here will be released, the mRNA will be released, and the ribosome will be broken apart or basically dissociated and ready to go again. We'll get into a little bit more detail in each of those, but this slide is a great summary slide to tell you exactly what goes on in the whole process of translation. So, like I said, the very first part is that initiation stage. At this point, you'd have an mRNA, the initiator tRNA, right? This is the one that has the anticodon to the start codon. And then we'd have the ribosomal subunits that are getting ready to associate into that initiation complex. In bacterial cells, we'd have 3 initiation factors that play a role as well. They're just termed initiation factor 1, 2, and 3. And it's that the initiator tRNA that recognizes the start codon in mRNA. In bacteria, this tRNA is designated as tRNA FMET. This is actually a modified methionine. It actually has a N-formal methionine is what it is. So it's slightly different than what we would see in eukaryotes. So just a covalently modified methionine. That start codon, though, is still AUG generally. In some cases, it could be GUG or UUG, but in general, you'll often see it shown as AUG. That first amino acid, then, of your polypeptide is always going to be the N-formal methionine. Okay, and that's in bacteria. What we'll see is that the binding of that mRNA, in order for it to get started in this translation, it actually will have to bind to part of the ribosome. And in this case, the ribosome has not formed a complete ribosome with both subunits. But what you'll see is that we have our mRNA. Hopefully you remember the Shine-Dalgarno sequence that is upstream from our start site. This will actually have a complementary portion of our RNA in the 16S rRNA, which is the part of the smaller subunit, that is complementary. It's actually this binding here that will facilitate the start of translation as well. So we can see that this part of the rRNA, the ribosomal RNA, is actually part of the 30S subunit, so the smaller subunit, and we will get some hydrogen bonding here, which actually attaches our mRNA to one of our subunits of our ribosome. You can see here that this is showing that 30S subunit. In this case, it's kind of the bottom of the A, P, and E sites. We have our initiation factors that would be found in these. Along with this section that is complementary to the Shine-Dalgarno region. What ends up happening then is our mRNA will bind here. You can see that that Shine-Dalgarno sequence will bind to that portion that is complementary. We then have that start codon as it's snaking through will end up in the P site. This is going to be when we're ready to get translation initiated. And so at this point, the initiation factor 2 will actually promote the binding of that initiator tRNA to the start codon in the P site. It's going to be in our next slide. You can see then that we have that IF2. It actually will help bring in that tRNA that matches our start codon. And then at that point, we will lose our initiation factors, and the 50S subunit will come in and combine, and we now have our complete ribosome. Again, this is the very first tRNA that comes in. It has that modified methionine, and it has the anticodon complementary to the start codon. This is showing over here the Shine-Dalgarno region with the area that is complementary with that smaller subunit. That would be the completion, basically, of that initiation of translation. In this case, that charged tRNA will enter at the P site. This is the only tRNA that actually enters at the P site. From this point on, all the rest will come in at the A site and then have the amino acid attached to it from there. So only the first one will start at that P site. And once that 70S initiation complex comes together, so the larger subunit of the ribosome comes in to bind with the smaller subunit, this ends the initiation stage of translation. Translation then goes into, translation is slightly different with the eukaryotes. The assembly will be similar to that, but there will be way more factors that that are required for this. Instead of calling them just initiation factors, the IF, we put a small e in front to note that it is a eukaryotic initiation factors. And this has a long list of them. Don't worry, I don't expect you to know them, but just know that it is a more complex start to translation in eukaryotes than in prokaryotes or bacteria. So the initiator, tRNA in this case is tRNA met, and that's because it carries methionine, and this is different than bacteria that carries that formal methionine. The start codon is this AUG, and it's generally speaking the first AUG on the mRNA after that 5' cap that our mRNA has been modified with after transcription. This is just showing the consensus sequence upstream from that start codon. And these can play a role in that start codon being recognized kind of optimally. The most critical nucleotides have their positions in red. So you can see here at the upstream of -3, we have our start codon, and then the one after the start codon. The rules for this are actually called the Kozak's rules, and this is after Marilyn Kozak, who proposed that this generally is what we see in the most optimum conditions for translation. So translation initiation in eukaryotes is fairly similar to bacteria. You can see we have our initiation factor complex will bind to that 5' cap in mRNA, so it's a little bit different. It's not the Shine-Dalgarno. And then the mRNA binds to a complex that is the small subunit, obviously the first tRNA and other initiation factors. Then that entire assembly will move along the messenger RNA looking for the start codon. Once it finds that AUG or start codon, that 40S subunit will bind to it, and we have the larger subunit that will bind as well, and this would be the start of initiation. That 80S then, ribosome is ready to go, and we have initiation finished. Translation into the elongation stage. During this stage, the amino acids are added to the polypeptide chain one at a time, and we see that the addition of each of these amino acids will occur through a series of steps, and these are outlined in 15.17, which I will be getting to in just a second. This process is complex, but actually happens quite quickly. In bacteria, it's 15 to 18 amino acids per second, and in eukaryotes, it's 6 amino acids per second, so quite quick. I think I'm going to pause here. My mini lectures only go 15 minutes, and I don't want to rush through this next part. So I'll pick it up in just a second with the last part of 15.7, I think we're on. 15.6, sorry. If you have any questions, let me know, but I'll pick up on slide 48 when I come back.